Hey y'all, Grandma Rose here. Today I'm going to talk about Roundup. Now the past two videos I've done in this series, the first one was about hybrids. What are hybrids and how are hybrids created? And then the next video, part 2 of the series, was about GMOs. What are GMO plants? And how are GMOs created? So I think it follows, let's talk about Roundup. And maybe in the next video I'll talk about BT, about the insect resistance. I don't know a whole lot about the virus resistance or about anything else, but I do know a little bit about Roundup and do a little bit about BT. That one probably will be short because there's not a whole lot to tell about that one. Roundup is a different story altogether. Let's talk about that. So, the question is, what is Roundup? How was it discovered? When was it discovered? All of these things. And what is it? What are Roundup Ready seeds? How are the seeds or the plants tolerant to Roundup? I'll tell you what Roundup does, and then we'll work on how are these plants tolerant to it. Let's get on with that. Okay, let me get my notes here, because I did take some, and I don't want to forget anything. Looking down, getting my notes and putting them in my lap, so maybe I can get them. Okay, so let's talk about this. What is Roundup? Well, Roundup is the trade name, Monsanto's trade name for the chemical glyphosate. What is glyphosate? Okay, what glyphosate is a modification of the amino acid glycine. Now there are approximately 20 to 22 amino acids. When I took biochemistry, I think I learned 20 to 21, and since then there has been an additional one that's been discovered. So as far as right now, I believe the count is 22, and who knows, there may be more that are discovered at some point. When I learned about it, there were 21, now there's 22. Now, amino acids are building blocks of proteins. So let's think about this. The way I think about it, and how they're made and how amino acids build proteins, I think about the DNA molecule. And you think about a DNA molecule looks like railroad tracks that are twisted. I think about a zipper. And a zipper's a good thing to think about too. And I think about on the railroad tracks like a hand car. You know, one of these hand cars that somebody cranks the lever up and down and makes it go down the railroad tracks. And as it's going down the tracks, it's reading what each of the bars along the way codes for a different amino acid. And when I think about this, what I thought about just a week or so ago. Jess of Roots and Refuge Farm visited Baker Creek Seed Company in Mansfield, Missouri. And as part of that, they looked at like the assembly line. I mean, it wasn't an assembly line per se, but it was filling orders. Somehow or another in that line, a light would shine on the box where the packet of seeds was. And the worker who was filling the order would reach over and they would get the package out of the box and add it to the order. And then the light would shine on the next box and it would get that package and add it to the order, etc., etc., etc. And that reminded me of building a protein. And that's pretty much what happens. There is an RNA that reads the DNA. So there are like three of the bases in the DNA molecule. The three of them together code for an amino acid. And then this RNA particle is attracted to whatever the amino acid that codes for and adds it on. It tacks it on the end until eventually the protein is assembled. That really is how it happens. And I really think about the guys in an assembly line tacking on and building in block by block by block by block until the protein is finished. Now, some of these protein molecules, well, not some of them, all of them fold on themselves, and they create like a massive spaghetti, if you think about it. It forms a three-dimensional structure. And that amino acid, the amino acids in that then, that make up the protein, will bind to certain sites on a cell membrane, or a membrane within the cell, not necessarily a nuclear membrane or the outer membrane, but all of the different organelles within the cell have a membrane around them. And so the proteins are attracted to whatever site it is they're supposed to go to, and like a lock and key. So you think about a docking port, and that's exactly what it is. It's like a docking port. And the protein will have, say, like a key, and in the membrane there'll be another protein that would be like the lock. And that key exactly matches that lock, and only that key will open that lock. So that's how Roundup works. There's a pathway in cells, only in prokaryotic cells. Now, do you know the difference between a prokaryotic cell and a eukaryotic cell? Okay, prokaryotic comes from Greek words, pro meaning before, and karyon meaning kernel. So before kernel, meaning before a nucleus, considering the nucleus in a cell was a kernel. So a prokaryotic cell means before the nucleus. So that means cells that are prokaryotic cells don't have nuclei in them. Their DNA it's like a mass of spaghetti in there and it's not contained within a nucleus. Now in these cells there's a pathway. Let's back up here. Do you understand what a pathway is? I mean, I think we probably all understand what a pathway is. A pathway is like a series of steps leading to something. So you can have a pathway through the woods. You can have a pathway to a field leading to town. You can have a pathway through your garden. But essentially what a pathway is a series of steps leading to something. So in cells, what it means, or in chemistry, what it means is it's a series of steps, not steps as in stepstones, but as stages. And each one of these stages is like a precursor of the next. In a chemical pathway, you have a series of steps leading to the formation of something. And that's what this pathway is. And it leads to an enzyme. And what happens is this enzyme is like the key that opens the lock in this pathway. And without the key, then that lock stays closed and the pathway doesn't function. And so the enzyme will open that lock, and then the pathway becomes open, and when that happens, then there are three very, very essential amino acids that are produced. Now this happens only in very, very primitive groups. It happens in prokaryotic cells, which means cells that don't have nuclei, some fungi, and in the chloroplast of plants. Now why in the world would it be in the chloroplast of plants but not in other organisms? Well, we don't know about that. It's speculated, and this has been a speculation for a long time, was that plant chloroplasts originated as individual algae. And somehow or another the algae were engulfed by another cell and were happy. And the cell and the algae live in symbiosis. And in time, the algal cell within that organism became essential for that organism's life. It produced the sugars, it produced things that the other part of the cell required to live. And so it became fundamental to the life of that organism. And if that's the case, and it's not known for certain that that is the case, but it's speculated that that's the case. And so that's why the chloroplasts within plants could be related to these other prokaryotic cells. But this pathway only exists in these very, very primitive organisms, including cell chloroplasts. It's not included in higher organisms. And a eukaryotic cell, which are the cells in higher organisms that have membranes around their nucleus, they have different organelles within the cells, and they all have membranes around them. And those are called eukaryotic cells. Not sure what the prefix "eu" means, but I would imagine after or containing or whatever. So, in other words, it has a karyon. It has the kernel in it. So, that's how this pathway works. And because this enzyme unlocks the key to the pathway, it matches up with the docking port on it. That makes the pathway function. And these amino acids are produced. Well, what happens with Roundup, the glyphosate molecule is so similar to that enzyme, the docking port, or at least part of the conformation of the Roundup molecule is so similar that it binds to the same docking port and it blocks the enzyme. And when it blocks the enzyme, then that means that pathway can't function. The plant does not develop these enzymes, or the plant does not develop these amino acids, and it dies. It starves to death because it doesn't have these amino acids. So the plant starves to death because these amino acids that are fundamental to its growth and life are not produced. So that's really, really important, and that's how Roundup works. Or that's how glyphosate works. Roundup is just one of the forms of glyphosate. It was the first one. Let's talk about glyphosate and what is glyphosate. Now, I mentioned glyphosate is a modification of the amino acid glycine. Glycine is the smallest amino acid. And an amino acid, like I said, it's a basic component of proteins. Always contains a nitrogen group. That's what amine means it has a nitrogen in it. Things that have nitrogen in it. Ammonium nitrate fertilizer, obviously nitrogen. Ammonia, nitrogen. Urine, nitrogen. Have you ever had babies that had cloth diapers? Probably not. But my babies were cloth diaper babies. And if you put those diapers in a diaper pail, in a day or so they smelled like ammonia, really, really stinky. A cat box, stinky, ammonia smelling, if it's not changed as it should be. So, these amine, ammonia, whatever, stands for nitrogen, or it means that something has a nitrogen in it. That's going to be important later on. Maybe not in this video, but we're going to talk about that again. We'll talk about that again. But therefore, all proteins contain nitrogen. And there we are. That's why nitrogen fertilizers are so important in plants, and why nitrogen fertilizers make plants grow. Because nitrogen is essential. Every protein in plants, humans, any organism contains nitrogen, and without nitrogen you're going to die. And that's what happens. Okay, let's talk about glycine. Let's talk about glyphosate. So glyphosate is the chemical name of the trade name Roundup. Now I looked into this to see, I mentioned at one point not really understanding what was glyphosate, when was it discovered, and all of this. Well, there are contradictory reports on the internet. You know how the internet is. You can't believe everything you read on the internet. A lot of you have to pick and choose and be educated and understand, well, what's bunk and what isn't. So it took some work, but I discovered some websites say that there was a chemist working for the Monsanto Corporation that discovered glyphosate in 1970. and then it was patented subsequently. But when I looked some more, but it said he discovered it. Well, how did he discover it? Well, I looked further, and then I read that there was earlier than that, 20 years earlier in 1950, there was a chemist working for another company in Switzerland. So he was a Swiss chemist, and I did not write down his name, but just take it for a fact. There was a Swiss chemist who was working for a corporation, and he synthesized glyphosate. And what happened was he added a phosphate group to the nitrogen atom within glycine, and that made it gly, as in glycine, and phosphate for the addition of the phosphate group. So glyphosate means glycine plus phosphate. So he made that in 1950, and then didn't know what it was good for, but he synthesized it. Well, 20 years later, there was a chemist working for the Monsanto Corporation, and he was looking for water softener. And in looking for water softener, he discovered glyphosate. And then discovered that glyphosate was a strong herbicide. Well, 4 years later, in 1974, Roundup was patented by Monsanto. So it held the patent on glyphosate, on Roundup, from 1970 to 2000. And in 2000, the patent expired. And now, 2018, there are, I don't know how many, I cannot tell you how many different companies now produce a compound that is glyphosate, but under different names. So no longer is Roundup the only player in the ballpark. There are several different Roundup type glyphosate herbicides on the market now, not just Roundup. And all of these GMO plants are resistant to this herbicide. Now, how did that happen? Okay. Now, I don't know what year this happened, and I should look this up, and I didn't. I remember looking it up, and I discovered what happened, but I didn't write down the year that it happened. At a chemical processing plant that was making Roundup, in one of the drains, there was a bacterium. It's called an agrobacterium that they discovered was resistant to Roundup. Now remember what I said before, that bacteria, these prokaryotic cells, are sensitive to Roundup. They have this pathway. But anyway, this EPSP pathway exists in bacteria. And this one particular agrobacterium, and I mentioned before in the previous video that agrobacterium are used in the molecular biology, in the genetic engineering, in transforming cells, making GMOs. The agrobacterium is used in producing GMOs. Well, this agrobacterium didn't die, and it should have. So that was sequenced, it was studied, and they discovered that there was a modification in that pathway, that the enzyme was missing something, or there was, I think it was like a single point mutation. There was one amino acid difference. So what's the big deal? What's the big deal? One amino acid, what can that do? Did you know, you've heard of the disease cystic fibrosis, but it is a terrible disease, and it results from a single point mutation. Now, why is that so bad? Well, think about proteins. Let's imagine that if you had magnets, and the purpose, what a protein does is that the different amino acids have different functions and different shapes and different properties. Like say on a magnet, you're going to have a positive end and a negative end. And if you put the two positive ends together, they're going to repel each other. If you put a negative end next to a positive end, they're going to attract each other. We've all played with magnets, and we know how that works. So what happens if what used to be a magnet that had a positive end on the outside, and then another protein. Imagine that this is an amino acid, and there was a positive charge on the outside. And then on another part of the molecule, there was an amino acid that had a negative charge on that side. Well, those negative and positive charges would be attracted to each other, and it might make them bind together and form a sphere or a circle or a tube. Well, what is it? Like an inner tube. And it would have that shape because the positive and the negative charges came together. Well, what if the magnet, one of those magnets, was replaced by something that had an opposite charge? What if the one that had the negative charge was swapped with one that had a positive charge, and no longer did they attract each other, but they repelled each other? That would be a single change, but instead of binding together and having this conformation like that, you would have a conformation like that, and it would be totally different by one single change. So this is sort of like what happened with this bacterium and the enzyme that it produced. There was a single change, but it changed the conformation. It changed the shape of the protein that was produced. Where before, Roundup was repelled by it, or Roundup was attracted to it. Roundup was attracted to it and would bind to it, and it inhibited the function of it. But the shape that it is now, with this one change, Roundup is no longer affected by Roundup because the shape of it is different. So let's say before, where it was blocking the opening to the gate, and the enzyme couldn't get in, with this other shape, the enzyme is able to get in. And it doesn't matter if Roundup is there or not, because it's immune to it, and it's not affected by it. And that's how a single point mutation can make a difference. And so scientists sequenced that gene, and they cloned it, and then they inserted it in the genome to form Roundup resistance, where that made that bacterium, that agrobacterium, resistant to Roundup. They were able to take that mutation from that bacterium and put it into plant cells to make the plant cells resistant to Roundup too. And we know what that led to. Okay, so people, animals, don't have this pathway. So why are people worried about it? Why should you be worried about it if you don't have it? Okay? Well, why should you be worried about it? Roundup isn't going to work on you. In fact, now this is crazy. We know about bacterial-resistant bacteria that have become antibiotic resistant. And when I was researching this, I discovered that there are some scientists who are actually looking at the possibility of using glyphosate to treat bacterial infections. The bacteria that have become resistant to antibacterial drugs would still be susceptible to glyphosate. It would block this pathway, and they would die. So there are scientists who are actually working on and considering on using glyphosate as an antibacterial agent to treat infections. Now that's kind of crazy, isn't it? What do you think about that? I don't know. What do you think about that? Let's think about this some more. I want to talk about one more thing. Let's think about something else. Talking about blocking, not producing an amino acid. Do you remember in the movie or in the book Jurassic Park, the scientist who engineered the dinosaurs in Jurassic Park engineered in them a lack of an amino acid, and I don't remember which amino acid it was. And the only way they could get that amino acid was through the food that the scientists were feeding them. Without that food, then they would die. Well, do you remember in the movie, at the end of the movie, the dinosaurs escaped, and they went to the mainland, where they fed or they feasted on soybeans and other crops that contained that amino acid that they required, and they were able to survive without the supplemental food that the scientists were giving them. So that's the same kind of thing. Without that amino acid, those dinosaurs would have died. And without the essential amino acids that are produced through this pathway that Roundup blocks, plants die. And that's how that works. So why are people concerned?